Biological systems contain a highly complex array of different sugar polymers. Now, despite the complexity of these sugar polymers, our individual sugar monomers are actually quite simple. So how is this possible? How do we link simple monomers together in order to create more complex polymers? The answer is found in the reactivity of our carbonyl and alcohol functional groups on each sugar monomer. So again, as we've learned earlier, our alcohol and carbonyls can react together to cyclize our sugars and create this hemiacetal functional group. Or, of course, for ketoses, we would form a hemiketal. Our hemiacetal, of course, can always be identified as our carbon that has two bonds to oxygens. One will always be an oxygen linking our sugar into this ring shape, while the other oxygen will either be a hydroxy group or we'll have additional carbons attached on here for our hemiketal. Now, as we know from organic chemistry, our hemiacetals can continue to react with additional alcohols to form a full acetal or a full ketal in the case of our hemiketal. It's this reaction to form our acetal or ketal that is key for linking our sugars together. This is a reversible condensation or hydrolysis reaction, which this in and of itself is quite important for our sugar chemistry because it allows us to combine sugars together when we want to store them, but we can also break off individual sugar units and use them for energy in our body. So we can join essentially any two sugar molecules together using the hemiacetal or hemiketal on our anomeric carbon and any of our hydroxyl carbons. This forms what we call our glycosidic bond. So this is essentially the equivalent of the peptide bond that we saw earlier for our proteins. Unlike our proteins, with our sugars, we have a wide variety of different glycosidic bonds that we can form, right? So our hydroxyl groups are found all over our sugars, right? We have multiple hydroxyl groups, which then means that we have multiple possibilities for bonding. So if we look at the sugar here, so we've combined together galactose and glucose to form a disaccharide. So this particular disaccharide is lactose. And you'll notice we've connected our first carbon, which is our anomeric carbon, from our galactose to the fourth carbon of our glucose. Now, again, this could attach to any of our other alcohols here, right? We could attach the alcohol at the third carbon. We have one at the second carbon. We have, of course, our anomeric hydroxyl group on the first carbon, and we also have our OH hydroxyl group on the sixth carbon. So in order to indicate where our bond is forming, we're going to use a nomenclature to indicate the positions of our reacting groups. So you'll notice in the full name down here, we have 1 to 4 to indicate the position of that glycosidic bond. We also have beta here representing that our galactose is in the beta anomeric conformation. Remember, of course, beta refers to when it is cis with respect to our terminal CH2OH. So rather than writing out this full complex name here, we can represent our disaccharide with just this simple nomenclature here. A 3-letter code representing our galactose, a 3-letter code representing our glucose, and the key for our glycosidic bond is 2 numbers to indicate the positions linked together, as well as any necessary alpha or beta to indicate the form of our anomer. Now again, there are lots of different possibilities for where we can connect our sugars. So we can connect it to any of our hydroxyl groups, including the hydroxyl group on our 2 anomeric carbons. So here we have 2 possibilities where our sucrose is a disaccharide formed linking our first and second carbons of glucose and fructose, which happens to be both of our anomeric carbons. So we've linked these 2 together to form a full acetal and a full ketyl. Of course, the fructose over here has our ketyl, since it is a ketose sugar. So in this case, we have to change our naming to include alpha 1 and beta 2 to indicate the anomers of both of these anomeric carbons. You'll also notice that our arrow here is double-headed. Normally, the arrow is going from the anomeric carbon to the hydroxyl group, but in this case, they are both anomeric carbons. Here's another possibility down here in our trehalose disaccharide. In this case, we've linked two carbon 1s together, our anomeric carbon from glucose, to form an alpha 1 disaccharide. Now you might notice for both of these sugars, we've eliminated all of our hemiacetyls and hemiketyls. So at this point, these are non-reducing sugars. So our reducing ends, of course, are those reactive hemiacetyl or hemiketyl ends. And in this case, we've used both of our hemiacetyls and hemiketyls to form our nucleic acidic bond. And we've created a sugar then that is much less reactive. So these non-reducing disaccharides are relatively common. They tend to be less reactive sugars overall, although you could still have other sugars with reducing ends that might react to a sugar that has no reducing ends. Our polysaccharides will expand upon this disaccharide structure to create much larger sugar polymers. So again, we're going to be linking those hemiacetyls or hemiketyls to hydroxy groups, and we'll just continue to repeat this to create very large polymeric structures. Now, there are many different ways to classify our polysaccharides. So they can be either homo or heteropolysaccharides. So a homopolysaccharide would just have one single repeating monomeric unit, whereas a heteropolysaccharide would have multiple different monomeric units, right? So a homopolysaccharide might just have glucose, for example, linked together again and again, whereas a heteropolysaccharide will usually have two different sugars, although it can have even more unique sugars. Our polysaccharides can also be either linear or branched. So a linear polysaccharide would have just one type of glycosidic bond repeated over and over again. So this would be similar to our proteins, right? Our proteins are linear chains of amino acids. But we can also have branched polysaccharides. So with our sugars, since they can react between our anomeric carbon and any hydroxyl group on our sugar, we can create a wide variety of different kinds of glycosidic bonds. And we can potentially have a polymer then made with multiple different types of glycosidic bonds to allow for different branches within our structure. So this already will create some additional complexity compared to our proteins, where in proteins you only have one possible bonding site. Here we have multiple bonding sites. In most cases, our polysaccharides will not have a defined molecular weight. So in contrast to our proteins, our polysaccharides do not use a template for their production. So they'll often be more in a state of flux, where we can add or remove our monomeric units as needed, right? So we could take off some sugars that we want to use for energy, or we could add more sugars when we have excess energy and want to try to store that. So here we see representations of our different kinds of polysaccharides. So we see our unbranched or linear, which can be either homo or heteropolysaccharides. Note the different colors here are representing different kinds of sugars. And then we can also have sugars that have multiple glycosidic bonds linked to them, creating branching points to make our branched polysaccharides. Now, as we're creating these large sugar polymers, so we are going to be using different kinds of sugar monomers, so it's important that we have a system for identifying what sugars are present within our larger polymer. So we're going to use abbreviations similar to our amino acids. So our sugars will have 3-letter abbreviations. So these will generally be the first 3 letters of the name, but it will change, of course, depending on what kind of sugar we have. So note for glucose, we use GLC, since of course the GLU abbreviation is already used for glutamate. Now, in addition to our 3-letter codes, we also commonly use colored symbols to represent our different sugars. So you'll note we have different sugars for different monosaccharides, yellow here for galactose, blue for glucose, and green for mannose. And then our modified forms of those same sugars will use the same color, but a different shape. So we have a yellow square for the N-acetyl galactosamine. So this is one of those amine-modified galactose sugars. We can use the same square but a different color to represent our glucose with the same N-acetyl modification, and so on and so forth, right? Notice the same modification will always have the same basic shape with just the color change to represent the sugar. Now, it's not critical that you memorize all of these because we won't really look at a ton of different sugar varieties in our polysaccharides. Now that we understand the basics of our polysaccharides, I want to take a closer look at the structure and function of some of our common polysaccharides. So we'll start with glycogen. Glycogen is a branched homopolysaccharide of glucose. So of course, branch should tell you right away that we're going to have multiple different glycosidic bonds here. And the fact that it is a homopolysaccharide will indicate that we just have a single sugar, in this case, glucose. So our glycogen is formed from glucose linked together with alpha 1 to 4 glycosidic bonds. So again, the alpha refers to the fact that we have our anomeric carbon linked in the alpha anomer to the 4th carbon with its hydroxyl group between our 2 sugars. So we'll use this alpha 1 to 4 bond to create very long linear chains. And then we'll create occasional branches with alpha 1 to 6 linkers. So these will occur roughly every 8 to 12 residues. So the end result, again, will have kind of long chains of sugars and then occasional branches coming out, creating new long chains. The overall molecular weight of our glycogen will often end up being in the millions. So we'll just keep on attaching sugars as long as we want to create a larger and larger storage polymer. So our main function of glycogen is to store sugars in animals. So this includes in our body as well. So we can link that glucose together, and then we can pop off individual glucose molecules when we want to metabolize those for energy. In contrast to glycogen, starch will be used for storage of sugars in plants. So starch is also a homopolysaccharide, but it's a mixture of 2 different homopolysaccharides together. Again, using glucose as our primary unit here. So we have one polysaccharide of amylose, which is unbranched. So this would be a linear polysaccharide with only a single glycosidic bond, which is our alpha 1 to 4 linkage, just as we used for glycogen. So that will look very similar to glycogen, but without the branching. And then we'll combine that with amylopectin, which is almost identical to glycogen. So it's a branched homopolysaccharide, but our alpha 1 to 6 branches will only occur every 24 to 30 residues. So it will be similar to glycogen, but with less frequent branching. And again, we can create molecular weights into the hundreds of millions as we link hundreds of sugars together to create these large polymers. And again, as I mentioned, starch will be our main sugar storage source in plants. So starch we'll use in plants to store our sugars, and glycogen will be used in animals. So here we can see a closer look at the structure of our starch. And of course, our glycogen as well will have nearly identical structure. Of course, our key monomers here are always the same, since these are our homopolysaccharides of glucose. Glucose we can recognize by the unique pattern of our chiral centers around our sugar ring. So we always end up with kind of this alternating pattern of our hydroxyl groups. So here are the alpha 1 to 4 linkages that create the linear chains of our amylose, our amylopectin, and glycogen. And this is what our alpha 1 to 6 branches will look like for our amylopectin and glycogen. So notice with that alpha 1 to 6 glycosidic bond, we're taking the anomeric carbon of one glucose and connecting it to the terminal CH2OH of another glucose molecule. Notice that the molecule that has our alpha 1 to 6 branch will still have the standard alpha 1 to 4 branch, or alpha 1 to 4 connector rather. So you'll then just have two glycosidic bonds to the same sugar. Again, the structure of glycogen will be nearly identical to the structure of amylopectin shown here. The only difference would be the increased branch frequency of our glycogen. So the overall structure of our polysaccharides will look something like this. We have very long chains of our sugars, kind of shown here as almost like a spiral. We have these occasional branching points represented in red. So these are the alpha 1 to 6 glycosidic linkages, creating new branches off of our main chain for our amylopectin. In the case of starch, we'll also have our amylose linear polymer kind of mixed in here and integrated with our amylopectin. Now, it is important to note that since we are using all of our anomeric carbons to link onto our sugars here, we're only going to have one free anomeric carbon at each end of our sugar polymers. So these are the reducing ends. We have one for amylose and one for amylopectin. But of course, we have a wide variety of non-reducing ends, one for each branching point on our polymer. So amylopectin has a whole bunch of different non-reducing ends, whereas our amylose only has a single non-reducing end. This is kind of like the N versus the C terminus of our protein, except in this case, we can have multiple non-reducing ends if we have a branched polysaccharide. Because of the large polymeric structure of our glycogen and starch, they'll tend to be insoluble in water. So rather than dissolving freely in our cytosol, they'll tend to kind of clump together in granules within our cells. These granules will then also have specific enzymes that will be associated with our polymers and will help to either synthesize and add on additional sugars or pull off sugars and degrade these polymers depending on the energy needs of the cells. As we just noted, they'll have one reducing end and many non-reducing ends based on the branches. And so normally our enzymes will actually react at the non-reducing ends so that we can have many different enzymes working at once. And we can potentially cut off a bunch of sugar at the same time if we want to kind of rapidly produce sugar from our polysaccharide. So that would be much faster than having one enzyme working at the reducing end, since we only have one unique reducing end for each sugar polymer. The next polymer that we'll look at is cellulose. Now, cellulose is also a homopolysaccharide of glucose, but it's a linear polysaccharide. So it's more similar to amylose than glycogen or amylopectin. Now, unlike our amylose, our cellulose links together with beta 1 to 4 glycosidic bonds. This small change in our glycosidic linkage will actually create a significant difference in the behavior of our cellulose polymer. So cellulose is the most abundant polysaccharide in nature. It's found in a wide variety of different fibrous structures within plants. Now, one of the interesting things about our cellulose is that our beta 1 to 4 linkage actually changes the interactions between our sugar units. So we end up with our sugar structures forming a lot of intermolecular hydrogen bonds between our adjacent sugar monomers. These hydrogen bonds can also form between adjacent chains of our sugar polymer. So notice here we have those hydrogen bonds that I just illustrated to you between our monomers, but we can also kind of lay our sugars next to each other and form hydrogen bonds between our different polymeric chains. This high degree of hydrogen bonding between our sugars and cellulose creates a relatively tough structure, right? So these will combine together to form the fibrous structure of our cellulose polymer overall. Now, because of this more fibrous structure, as well as the insolubility of cellulose due to its large size, it's actually a relatively tough structure and difficult to react with for our enzymes. So most animals cannot use cellulose as a fuel source, so we can't ourselves, because we lack the enzymes that we need to actually hydrolyze those tough beta 1 to 4 linkages. Now, of course, there are some organisms that do have a cellulase enzyme that can cleave our beta 1 to 4 linkages in cellulose. So these will then be able to use wood or other plant material that's tough that includes that cellulose polymer as a source of sugar and as a fuel source ultimately. This difference between our cellulose and our starch or glycogen is partially due to the 3D structure of our polymer. So I want to briefly look at the overall 3D structure of our sugars in addition to just looking at the connectivity between our individual sugar molecules. So when we're looking at our polysaccharides, we also have dihedral angles that we want to consider, just like our proteins. So we can essentially have a phi and psi angle on either side of our glycosidic bond that will determine the overall shape of our sugar polysaccharide. And again, between an alpha versus a beta glycosidic bond, we'll have a difference in how those dihedral angles can be arranged. So our sugars will actually have a Ramachandran plot similar to our proteins. So here we see a very similar plot with our phi and psi angles, but in this case, this is represented with a 3D view here. We'll have some areas that are unfavorable, represented by these high red peaks here, and some areas that are more favorable, represented by our blue spot and these valleys here. And again, just like our proteins, we're going to be looking at the same key factors here for determining our dihedral angles. Right? It's going to be where do we have steric hindrance, and where can we form hydrogen bonds between our individual sugar units. So it's very similar in concept to our original protein Ramachandran plot. So for our sugars, you'll notice we want to tend to avoid very high angles that will end up with our sugars kind of bending back on themselves and creating that steric hindrance here. And we'll tend to like more extended angles where we can spread out our sugars and avoid that steric hindrance. When we compare the 3D structure of starch versus cellulose, we notice some interesting differences. So just by changing our linkage here to our alpha form, we tend to form a more compact structure for our starch. So you'll notice we have a fairly sharp angle here between our two sugars, and we can then take these sugars and form them into a helical structure. So we get a helix kind of similar to the alpha helix that we see in proteins, although in this case our helix is much wider compared to that alpha helix. So you'll notice we require 6 sugars to complete a full turn of our sugar helix here, whereas in proteins it was a much tighter helix with only 3.6 amino acids per turn. So our starch will tend to form these sort of helical structures, and will actually end up winding together with an additional helix to form a double helix. So usually when you hear double helix, you tend to think of DNA, but we actually have a somewhat similar double helical structure for our sugars as well. So our starch double helices will then end up kind of packing together into these tight structures here, although we do have water channels kind of available here as well, where water will be able to access and hydrogen bond to our individual sugar molecules. So now if we go back to our original drawing of starch, you can notice why we have our helical structure here of our branches, right? So these helices are actually double helical structures formed from winding multiple sugar polymers together. And this will also allow us to combine our amylose with our amylopectin in a relatively tight, compact structure, right? So the amylose is actually wound together in our double helix. So starch ends up forming a helical structure somewhat similar to the alpha helices in proteins. Interestingly, our cellulose arranges itself in a sheet-like structure, kind of like our protein beta sheets. So with our beta 1 to 4 linkage, we tend to form very extended structures instead of the more compact and turning structures of our starch. And so our most stable conformation ends up being laying our sugars flat and next to each other so that they can hydrogen bond. We can even stack them on top of each other and form essentially sheet-like conformations like our beta sheets. Now, this structure here might remind you of the silk fibrin protein that we saw earlier, where we can lay all these sheets on top of each other and create very strong fibers with all the hydrogen bonding and interactions between these different sheet structures. So again, this creates the very tough, fibrous nature of our cellulose, whereas our starch and glycogen with the more helical nature are more accessible for water and for enzymes then to react and break apart that sugar. So even a very small change, just in switching our configuration from alpha to beta, can end up creating a very large 3D structural change that then completely changes the function of our polymer. Next, I want to take a look at some polymers that are formed from other sugars. So first, we'll look quickly at chitin. So chitin is a linear homopolysaccharide, but it's formed from N-acetyl-glucosamine. Now, as I mentioned earlier, N-acetyl groups are quite common in structural polymers. So they slightly change the reactivity of our sugars. And in this case, we're going to use a beta 1 to 4 linkage between our N-acetyl-glucosamine monomers. So this is the same linkage that we had in cellulose. And so it ends up forming extended fibers that are very similar to cellulose. So we end up with a very hard, insoluble structure that can't be easily digested, which makes it great for forming tough structures in mushrooms or in the exoskeletons of insects, spiders, crabs, and other arthropods. So the chitin makes kind of a tough coating that you might see on things like beetles that helps to protect the beetle from their environment. The last class of sugar polymers that I want to look at is our glycosaminoglycans. So these are the first heteropolysaccharides that we've seen. So they will have repeating disaccharide units. So we'll have two unique sugars linked together in a disaccharide, and then we'll repeat that over and over again to create our polymer. So these are our key structural polymers in the extracellular matrix in animals. They will also play a key signaling role throughout our body as well, which we'll look at that later. So they tend to have a higher negative charge density in order to enable them to carry out those different interactions, right? So we usually want stronger interactions if we're going to be acting as signaling molecules. So we're going to take a closer look at these sorts of signaling roles of sugars in our next video.